Hello to all of you. It's a pleasure to introduce Esteban Miglietta. During this Spanish Heritage Week, we've had, among other speakers, people with different career paths, people from different backgrounds within the general area of microscopy and biomicroscopy. This is the first time I've worked with Esteban. I worked with him when we were profilers. I was talking with one of our colleagues here about that experience, and she referred to the courses they organize. And Esteban's contribution is important in all of those. Esteban was born in Buenos Aires. He studied biology there, and then later he earned his PhD focusing on Parkinson's. That's where he developed an interest in the analysis of image analysis. And I heard about his work when I was earlier in my career. Now he is working as a postdoc at Harvard, MIT with Beth City. And within that context, we've been working at our partnership to improve we've worked together as profilers again. We've organized workshops, and we've gotten to know each other better through the BINA series. It's a pleasure to introduce him, and there's a link to a paper that we wrote together about the postdoc program in which he's active right now. So here we go.
Thank you so much, Mariana. I'm sharing screen. Can you see it well? Well, thank you very much. First and foremost, thanks to BINA for this space and to Noemi and Daniel, who are going to be interpreting today. It's really a luxury to be able to give this talk in my native language, which is Spanish. And as you can see, the slides are in English. So if at any point my wires get crossed or I get stuck, it's something natural because it's what happens when you're constantly working with two languages in your head. And so as Mariana said, my name is Esteban. I'm a postdoc at Cimini's lab with the bioimaging lab, which is a joint effort with Harvard and MIT. And so now I am going to share a little bit about my career path with you. And this chat is going to be, or is organized in three different sections. The first one is a somewhat personal review of my career. And then, as Mariana mentioned, I am going to share a little bit about the postdoc program in which I'm participating. And at the end, I'm going to give just a small sample of of a collab that I was part of. And as Mariana said, I am going to start by sharing about my career, my personal career, professional career. It's not what I really love doing, but when I have listened to other people talk about their own arc of their career, particularly with BINA, it has really been useful for me because it has helped me put in perspective my own path. And so I hope to do the same. And as was already mentioned, and perhaps you noticed by my accent, I'm from Argentina. I studied biology at a public university there. I am a graduate of the University of Buenos Aires, and I did my PhD mostly working for the Leloir Institute Foundation. And what I was studying for my PhD was inflammatory responses and neurodegeneration, and that were linked to a protein that is very prevalent in this disease by using animal models. The the PhD for me was a very difficult stage. There were a lot of frustrations, experiments that didn't work out, results that were not happening. And on top of everything, towards the end of my PhD, that's when we had the pandemic. So in total, it took me 7 years to complete my PhD. And at the end, I did not have any papers published as an author. Um, and so for that reason, I lost interest a little bit with regards to following a traditional academic career, and also in part because I thought that maybe I wasn't made for that. However, not everything was bad. There were some aspects of my PhD that I was really able to enjoy. Like, for example, the work that I did in collaboration with other folks, helping other folks, supporting them with their work, both within the lab as well with other groups, and also the work that I did in microscopy and bioimaging analysis of image analysis. And so one of the habitual tasks that I had under my charge because of the model of work was this to study the neurons, like in this image. So each of these small black dots, and I was doing this to evaluate animals where I was trying to express my mutated protein, and I was trying to see if there really was, through this special treatment, if there was neuron death as it happens in Parkinson's disease. At the lab, we had specialized equipment to carry out this cell counting task. As you can see, it's several, like 400 per cell. And so I had a stereotaxic microscope to do this efficiently. Unfortunately, by the time that I came into the lab, that microscope was broken, it wasn't working, and we did not have the resources to repair it. And so what we ended up doing was that we hand counted these neurons. So that was not my favorite situation, and not only because this is very tedious work, but it was a very closed experiment, and it wasn't it couldn't be reproduced. However, this turned out to be my favorite part of my thesis, because what ended up happening was that through a lot of trial and error, I was finally able to, by playing with an analysis software, particularly Fiji, I developed a very rudimentary method by using masking and very simple techniques. I developed and validated a semi-meta-analysis pipeline for counting. And so this was the halfway point between the manual count and this method. And so by doing this image analysis, it was very helpful because it also helped me develop a role within academia, where I was still in academia, but in a non-traditional role. So once I completed my PhD, I was able to work at the Leloir Foundation Institute under Dr. Petrosi and Dr. Pasquale, which is now under the leadership of Andres and Alejandra. This role was very important for me because it didn't just allow me to develop myself and to focus on those positive aspects of my PhD, which was collaboration and working with imaging, but it also allowed me to understand that my work had value and that I could either facilitate or even improve the work done by other folks. And so in this role, the work that I did didn't just include maintenance and technical support, but I also was training users. And above all, what really mattered is the scientific support with experiments, and at the end, even image processing and analysis, mostly. So back then I was already liking and was interested in image analysis. And so I leveraged this role to develop myself further by going to courses and reading the literature. And by doing that, I was able to collaborate with different groups within the Institute doing image analysis, mostly using using masking. And so because of this, and when I saw how valuable my work was to my collaborators, I thought that on one hand, there's a huge need in the scientific community to have um, trained folks, but this also meant a huge uh, opportunity for career growth. And so I decided that I was going to continue training in bioimaging, particularly image analysis. And that is how we get to the second part of this presentation, as Mariana mentioned at the beginning, the second part of this postdoctoral training program, which is very unique, in which I am participating. And because this is a BINA talk, I imagine that most of you are aware of the fundamental role that microscopy experiments play in life science research, as well as in the physical sciences. In addition, you probably also know that more and more, it is not enough to take a nice representative picture in the microscope, but it is necessary to quantify those images and turn them into numerical data that can be compared and can be reproduced. That is why, as I said before, there is a huge demand in the scientific ecosystem for professionals, both male and female scientists, that are trained in microscopy and image analysis. However, training opportunities, especially comprehensive training in these fields, are very limited. For that reason, in 2013, Jennifer Waters, who's the director of Harvard Medical School's Microscopy Center, and this, by the way, was recently renamed Core Imaging Technology and Education site. She launched the first training program for imaging scientists focused on training professionals who would later take charge of microscopy course in research centers and universities. To this pioneering program, later, another program was joined at the AIC Janelia, which is under the direction of Tenglong Chu. But as the technologies available for image data acquisition expanded and became more complex, so did the tools available for the computational analysis of these data. Today, there are more than 60 software packages available to the imaging science community. However, when these folks who use this survey, who use this software were surveyed, the survey showed that a high percentage of them don't feel like 100% comfortable with these computational tools. Like, for example, they don't feel comfortable writing code or using command lines. And so these are skills that these tools require. And so it was in this context that it became even more relevant to develop a new profile within the scientific ecosystem, which is that of the bioimaging analyst. And these are folks who come from different backgrounds, but who are at the intersection of biology, physics, and computer science or data science, which is where a lot of these imaging experiments take place. And so now these analysts can be the bridge between these disciplines. And so it was in this context that in 2019, and also in the spirit of replicating Jennifer Waters' program, that the bioimaging analysis training postdoctoral program was created to train these types of professionals. This program was created by Dr. N. Gardner and Beth Cimini, Dr. Beth Cimini, um, here at my institute, and I'm sure that many of you already know them. And both of them have a long history of creating and disseminating tools like CellProfiler. And Anne, as a matter of fact, is one of the creators. And Beth and her team are actually currently responsible for running that. And so, as I said, this program started in 2019, and during its 5 years of existence, 7 people have already gone through the program, and 6 others are in various stages of completion. Although all of us come from very diverse backgrounds, what we all have in common is that we are all PhDs with some degree of experimental science and use of microscopy. Um, and even though most of the work that we do is um, computational or uses computer, the use of computational tools is not a requirement, and our level of knowledge and proficiency with it really varies. In my case, I started the program with very rudimentary knowledge of programming, and one of my objectives in the laboratory is to improve this.
Program itself follows a curriculum that includes computational skills. We have various collaborations on scientific projects, all aspects of project management, because we have different stages going on in parallel, and then we engage in outreach. For example, we offer open consultancy or office hours to the general public. We reply to questions, remarks, and certain online forums, conduct workshops. This curriculum is not something that is rigidly structured. We try to respond to the needs of each different postdoc, each different participant, because in a survey that we ran recently in the study that Mariana shared, done with previous participants or alumni from this postdoc program, we worked with a series of different aspects of the training program and how important they were. What we were able to see at a glance is that for a lot of variation from person to person as far as what the most important aspect of the program was. In the next aspect of the curriculum, the program follows a hands-on learning sort of an approach, learn by doing. And so within industry and within academia, we are faced with a range of challenges, different ways of acquiring the images and different tools for answering the questions associated with those images. A lot of these programs, or the majority of these programs, are paid, which means that that income, the revenue helps to support the program. And so this establishes, as it were, protected time, or allows protected time in which you can follow your interests or needs that you identify, that postdocs identify their own. A particular or specific aspect of this program that we really like is the opportunity to interact among ourselves and with extremely experienced experts, as well as with other people, people from more senior cohorts within the program. Also, considering the fact that the lab is also devoted to producing tools for image analysis, and he listed the name of a few, it's a real privilege to be able to have access to and to interact with the individuals who are developing these tools or who created these tools that we so often use in our day-to-day work. We also have the opportunity for feedback. In the majority of cases, this interaction looks like this. They make a tool, we break the tool, and they fix it for us. The 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 program follows an interesting approach in that the approach on the specific tools and on image analysis have not been the most important factors in selecting successful candidates and predicting who these successful candidates would be. There are certain soft skills, such as the ability to communicate, organizational skills, but especially perseverance and the willingness or the drive to learn have determined who's going to do the best. This also means that it's extremely important to us to be able to talk or maintain within our lab, maintain a safe environment where people can formulate questions and pose those questions openly. There's a rule in our lab that isn't always followed, which says you should try to solve the problem on your own for a half hour, for 30 minutes. If you go beyond 30 minutes and you haven't solved it on your own, talk to somebody, seek help, ask questions. This reinforces the notion that this is an interdisciplinary work area, and in this kind of a work environment, nobody has all the answers. It also improves the efficiency of our work. We find the solutions more rapidly, and it establishes this cooperative environment. During or as part of this survey with the different cohorts, when we ask these people what other factors seem important to them or are important to them for their development within this program, the word cloud that we got, words that we see often are people, environment, culture, experience. Another aspect that is a key to all of this and that supports or is a central pillar to this is documentation, not just for the tools, the hardware and the software that we use, but these questions, regarding these questions that keep coming up. So we try to share these questions, express these questions in open communication tools such as Slack, so that the questions and the answers to these questions can be viewed by other people. So you don't have to keep asking the same questions over and over again, right? We like the FAQ approach. And we want people to be able to have access to that and to see that their contributions are. So part of the stipend or the salary for the postdocs it comes from external funding, and this can generate a certain tension between the need to be generating income for the lab on one hand, and on the other hand, the notion that you want your time to be yielding personal development growth or professional growth for the individuals. So this is important. That's why it's important to have this protected time for participants, for the postdocs. So you're not just machines for, we're not just robots making sure that paid projects get completed and milestones met. We have support from an NIH grant and from the Center for Open Bioimage Analysis. It's really important that we might to identify other sources of funding that we can tap into. I'd like to spend a little bit of time reviewing the positive effect that this program has had. On one hand, you can see this just by looking at the career paths of individuals who have graduated and where they've gone off into industry, into academia, the generation of new tools, new facilities, this variety of positions, data scientists, software engineers at startups, image analysts in universities. This is another example of the need for training and introducing professionals with this kind of profile into the market. This is not all entirely random. This didn't just show up. We have specific moments in spaces for professional communication. For instance, we have quarterly career chats during which we talk about your career path interests, and we start planning to see what we can do to reach those. And then there's one day a year, we have a career day once a year, and we devote the entire day to tasks related to professional development that may have been set aside during the rest of the year, such as putting your resume up to date or your LinkedIn page, find out about programs, funding sources that you can apply for. Something else that's interesting is to look at the community that this program has had on the community, the scientific community, and we can see this reflected in the numbers here. You see, for instance, 18 publications, many of which have to do with tools, software that are open to the community. We have organized 29 different workshops, 17 tutorial videos, as well as blog posts. We also, the office hours, the posts, I'm sorry, we're coming back now to the office hours, over 120 office hours open, and we have replied to over 2,000 questions and posts.
Having said all this, and from a really close position, I think it's important to continue to promote this type of programs and to replicate this in other places. And so to wrap up, or to the last part of the presentation, I'd like to give you, show you an example of a recently published project where I was a collaborator as an image analyst. And this is given in the current context in which advances in sequencing techniques have allowed the discovery of hundreds of thousands of genetic variants associated with various diseases of Mendelian inheritance. However, just in this decade, we have seen a lot of variation, a lot of variants which have been classified. However, the mechanism by which they negatively impact health is not yet known. And so that's why these variants that we see in yellow are known as variants of unknown significance. As a matter of fact, many of these variants that have already been identified as pathogenic, we don't understand yet how or in what way they impact health. But of course, it's important in order for us to find treatments or cures for these diseases. What's in our favor is that although the diversity of diseases associated with generic variants is enormous, and it affects a lot of tissues and stages of development, the underlying understanding of the mechanisms is very reduced. So let's say that the pathogenic variant has an altered protein, and the interaction of that protein is altered, or the localization is altered. In that case, what we can see is that this affects a certain percentage of the variants, but how that could be an underestimation. And so what could be affecting it? It could be ligands. And this is very difficult to predict. That is why to be able to classify this huge quantity of variants of unknown significance and to be able to find out what are the pathogenic mechanisms that lead to disease, it's important to study at a huge scale this through localization of proteins, and that is what we set out to do with this project. And to achieve a large-scale systematized analysis of protein localization in these variants, HeLa cells were transfected in 96 well plates with different genetic constructs that contain the gene of interest followed by a flag tag. We had nearly 3,500 variants that were studied, and they came from different libraries. And these variants were mostly cataloged as pathogenic, about 60% of them. And there were some recessive, some were dominant, and some were linked to the sex chromosome. Among these variants, 1,269 genes represented here as reference variant. And the most common alleles were also available. So after transfection, the cells were fixed and they were labeled with an anti-flag antibody to show the localization of the protein of interest. In addition, we marked the nucleus, the mitochondria, and the membrane and reticulum as references. We obtained images of these cells with confocal microscopes that had high throughput with a high resolution objective. And because we wanted to study delocalization in great detail through a tremendous human effort, we did it by hand, and it was done independently by two individuals, Jessica and Seal. And we studied each of these 8 compartments: the nucleus, the mitochondria, the membrane, and reticulum. And even though the notes from the two operators highly coincided, in order to be able to do this for so many variants, we realized that it wasn't sustainable in the future because we have thousands of variants to evaluate. For this reason, we sought to complement this human analysis with morphological profiling. So what am I talking about when I talk about image-based profiling or morphologic profiling? Well, in a quantitative typical experiment, what we do is we mark the protein of interest or the structure of interest within the cell, and then we do some measurements of that protein or structure based on a particular phenotype that we might be interested in studying. I don't know, it could be that we are studying the size. And then we compare those measurements under different conditions to find the answer to our biological question. But when we use image-based profiling, on the other hand, the technique is a bit different. What we do is we label the greatest number of organelles possible, and then we observe them under the different fluorescent channels. And so we use a profiler to do this. And through this huge quantity of data and of measurements that are quite agnostic, due to the biological question we have in each case, we really need image-based profiling to find answers. And so going into greater detail, here we use HeLa for the workflow. And so what we do is we subject them to different disruptions. We put them in multi-wells, and then we observe them, and then we analyze the images using a CellProfiler. And we study each cell and each nucleus individually. And as I said before, throughout all of these compartments and through every single channel, we carry out several measurements. And the set of all the measurements or measured characteristics of a cell constitute the morphological profile. This profile is basically a series of numerical values that characterize the phenotype of the cell. And so we compare the size, the markers, the intensity between markers, texture, and other things. And so when we compare, group, and classify each of these profiles, we're able to obtain interactions or determine what the interactions are. So that's how, to complement the manual count, we did systematic analysis of protein localization. And as I said before, we carried out a lot of measurements, actually 1,293. And based on these measurements, we filtered out all of the cells that had not been transfected, that were not, in other words, that were not expressing the protein of interest. And then we took 340 measurements in the green channel, where we can see that the protein is showing up. And this was done originally by Marce, and then I joined the team and continued this work. And so this could be an example where each line represents a cell, and all of the colors represent the different measurements. And as I said, all of these cells form the profile of a pathogenic variant. And so if we see the correlation between the pathogenic variant and the protein of reference, we will be able to find a value that gives us an idea of what is the impact of that variant over the protein localization. In this case, we can see that we have some value close to 0, which means that when the impact score is closer to 0, as opposed to when it's close to 1. Thanks to the fact that we had these two sets of data, we were able to determine that the impact score does provide information with regards to changes in localization of proteins, because proteins that are mislocalized, which are here in red, had a higher impact score than those variants that were not written down manually as mislocalized. And the impact score also indicated that not every transfected cell necessarily showed variation in localization. As we can see here, these 3 show it, but these other 3 in the periphery do not. So the question is, what proportion of cells showed the phenotype was also correlated to the impact score.
These two methods are clearly complementary. The computational analysis allows you to detect subtle differences that maybe aren't observable by the human eye. There are also other effects that might have been diluted in the impact score, but that we're able to determine here. Just to speak about some of the results that we've achieved, 250 confirmed mislocalized variants were identified, 11% of all of those that were detected, and those 250, these were found starting with all of those that were identified as mislocalized either manually or numerically, and then to repeat the assay. And there were these kits that were done that were required during the second round. In this image, we see that each line represents each of those 250 variants, and we're showing the reference location and where they wound up for the variant. So these lines show that the reference was located in the membrane, and for the variant, they wound up over here in the Golgi organelle. So in this diagram, we see that we identified some very significant changes in the localization for essentially all of the compartments that were studied, and we were able to. This mostly had to do with proteins related to the excretory result, and had to do with the cell skeleton, as it were. So these variations classified as pathogenic or mostly pathogenic, they had more changes and more significant changes in their localization than those of unknown significance. Also, in many cases, it was observed that the degree of change in this localization is related to the severity of the disease with which that mutation is associated. For example, we see with the Alexander disease, which is related to, did he say nutrients? In the GFAP variant, it's located in the intermediate filaments of the exoskeleton. And over here, there's same is true for the benign variant. However, for the pathogenic variants, what we see is there's a prevalence of diffuse marks here and inclusions in the within the cytoplasm. If we compare the different variations we looked at, the concentration of cells with the diffuse GFAP, this correlates to the age, the onset age for the each of these variants. So the degree of mislocalization relates to the severity of the disease. So as a result, this has given us the first large-scale map for the Mendelian what is this? This mislocalization is a common result of these pathogenic variations, and it affects roughly 1 in every 6 of these variants. We also noted that the mislocalization is particularly common in proteins that are related or that are involved in the secretory pathway. These results give us a foundation to use this type of experiment or assay to identify new for further discovery. The idea is to identify a small number of variants after scanning through all kinds of thousands of genes. Okay, so wide mutational scanning, that this can be related to deep mutational scanning that considers all the variations, but for just a single gene. But this live mutational scanning that we employed here is more easily scaled. It's more scalable using these different image analysis techniques that change in the location of certain proteins to identify common phenotypes. This is important for studying the hundreds of thousands of variants and to identify which of them are pathogenic. And this could lead to new experiments, new approaches that will identify and to classify these variants. In the spirit of what's just been described and as a continuation of this project or of this work, VERCHAMP is what arises. Variant characterization across the Mendelian proteome. So there are some folks who are already have been working for several years with the aim of characterizing 100,000 of these missense variants and 3,700 genes associated with this disease. How is this going to be done? There will be some high throughput studies and some studies in the interactions between the proteins, considering where we look at their abundance, using methods that are already described and widely used by collaborators. We will also analyze the location and the cellular morphology using these morphological profiling that are ready here. With all this, all that remains is to thank everyone, especially BINA, and to Vanessa, Nikki, Andres, and Mariana, who organized this event, Noemi and Daniel, for their work here, to the sources of funding, the entire team, all of the image analysis team, to the Leloir Institute in CONICET, with which I have been associated during my doctoral work, and the team here. Thank you very much. This has been a really great presentation. I've learned a lot of interesting things. Please do not hesitate in asking questions.
It always takes a while to build our confidence to ask a question, but in the meantime, I'm going to ask one. I think that it was during our last BINA meeting in Mexico. So I don't remember if it was the last one. I remember that um, it, that it was mentioned that segmentation should be easier. When it comes to accessibility, it should be as easy to do Excel, right? I remember that at some point using Excel, it was really difficult, and now everyone uses it. So how is the process in your own experience now that you are in that world? How easy is it to do that? I think that currently, at least in certain contexts, segmentation is now a problem that's been solved. Yes, clearly we still need to have some knowledge, but I think that above all, thanks to deep learning models like CellPost, it's actually really accessible. And I think that the methodologies are there. I think that we need now is for those methodologies to be more easily accessible to people. So, like for example, CellPost, it still requires installation in order to be able to benefit from it. So yes, I do think that in certain aspects, that's already been taken care of. It's a problem that's been resolved. But I think that now what's required is to use deep learning uh, methodologies to expand segmentation parameters. Otherwise, we will always the operator will always need to have certain knowledge of the image. Okay, we have a few minutes left. I do have more questions, but I want to first prioritize our audience. Does anyone have any questions? Victoria Repeto is asking in the chat, could you tell us a little bit more how to quantify the penetration of a phenotype or phenotype penetration? Well, in this particular case, penetration was evaluated manually. We were trying to determine what was the proportion of cells that were showing that mislocalized phenotype um, compared to the ones that were expressing the protein. It was not, this was not implemented here, but of course it all depends on what the phenotype is. But it would also be possible to do it through image profiling. And on this project and in many projects, the image analysis is not done cell by cell, but once you have the data from all the cells, you aggregate it to create a profile. But if you study the profile or the differences between cells, then you could isolate the different phenotypes and then determine how many cells. And in this case, how many of the transfected cells have the mislocalized phenotype, and then you can evaluate penetration. Diego raised his hand. Hello, everyone. Thank you, Esteban, for this talk. And I was just clicking on the links that were sharing the chat, and what really caught my attention, and I don't know if you were open in this Center for Bioimage Analysis project, because I'm really interested in it. And my question is really, if you have worked on a similar project, I don't know if it's related to what Mariana was asking. And the question is, how easy would it be to have protocols that are easy to use, that even someone who's not who doesn't have a specialty in image analysis can use, like, can, like, if we have like tons of images that we can download home and you follow a simple protocol to be able to process that data. I think that's very interesting. And the Center for Bioimaging Analysis, as far as I understand, does not have any projects like open source projects that people can access, but they do spend a lot of time generating protocols and tools. They do it through videos, through tutorials, and also in written form. Um, they do it though for more specialized audiences, like in the scientific community. Um, there may be someone who's a postdoc who has a tons of images and doesn't know what to do with them and is lost. So, who may be helped by this, but I don't know any projects that meet the characteristics that you were asking about, but I think it would be very interesting to have one. Yeah, I don't know if Zooniverse or something like that, that has uh, several. projects. I don't know if it's only for astronomy, but it's for, kind of open science in general. Well, I see that we are past time. I just wanted to say that I think that you're all doing incredible work. And one of the things that I wanted to mention is that is what you mentioned at the beginning, that you went from a PhD with very little experience with bioimaging to now being fully immersed in this world. I think that there's a value to that, you know, in and you had already said this is not for me. And so it's interesting that you made that jump. And so I wonder if there are young folks in the audience who are just starting their career and feel that way. And I also find the model that you're implementing very useful. Because you mentioned Jennifer's and Leon's programs. And it's interesting that it's an alternative to a traditional academic career, which is the career in facilities. And I think this is a great career. And I think that in the future, this will become very interesting too. So I just wanted to mention that. Yes, honestly, I think that these are career paths that are still being formed and developed. And so I wanted to make that clear and also to reinforce the fact that we are also doing science. We are scientists. Those of us who work in facilities are scientists, um, and or who do image analysis. We're just providing like a different perspective. Well, if there are no further questions, thank you so much. Esteban, it was a pleasure to listen to your talk and to hear what you're doing. And I'll turn it over to Nikki in case there are more announcements. Otherwise, we'll see you next week for Citlali's talk.